Okay, so we already have seen in the previous video the male reproductive organ. Now we will study about the female reproductive organ. Okay. Now the reproductive organ of the female is known as the Carpogonium. Okay. I have already shown you the image. No, it is just a Google picture only. I just Google it, but you will see like that only when you do the practicals. Okay. So let's see how it develops. It develops at the top of 2 to 5 celled carpogonial filament. Okay. Let's say this is the main axis. This is one filament. And from this top part, if this top part swell up to form a carpogonium, so this will be your carpogonial filament. Okay, this word carpogonial filament. It will develop on the female trichoblast. Remember, for the male antheridium, it is developed on top of the male trichoblast. So if it's the female, if it's the carpogonium, it will develop on top of the female trichoblast. Okay, means short filament, female short filament. Now, what about the shape? If you remember from that first slide, it looks like a cup, it looks like a flask. A flask is like how? Like a vase, no? It has a vase-like shape, no? Like a bulbous and then narrow opening at the top. It has a flask-shaped body, okay? And the base is swollen, which contains the egg, okay? And we have an upper elongated neck region. Remember, in Lycopodium, the Archegonium, it had these neck cells and the egg was here, no? So very similar to that. It is like a flask like this. Then this is the neck, which is little bit elongated. And here, this basal part will contain the egg. Okay. This is what I explained just now. Okay. So during the development of the Carpogonium, initially, remember we always start with one cell, which we'll call as the initial cell. So here also, even in Lycopodium, there was an initial cell. Here also, we will have one female trichoblast initial. Okay. There's always, you need one class leader, no? Like one class representative. I will tell the class representative, you inform the girls, will have class so-and-so time, or inform the girls to submit their assignment on so-and-so. Then she will be the initial, she will start and she will tell all of you, then you will do the work. Same, okay. This, it occurs everywhere, no? You need one initial. So a female trichoblast initial cell is developed on the central siphon, okay. Remember, we have this is a siphon, means the pipe, surrounded by the peripheral siphon, peripheral pipes. Okay. So, from the central part, a few cells, the female siphon, the female trichoblast initial will develop from the central part, the central siphon. And the location is just little bit below the apical, means little bit below the tip. This is the main axis. This is one trichoblast. This is the tip of the cells. Just maybe 1, 2, maybe from the third cell it will form. Okay. Here, this will be your initial. Then that initial cell, this initial cell, the brush, okay. This initial cell will then undergo division. It'll keep on dividing many times and will form the trichoblast, which has 5 to 7 cells. Okay, it will keep on dividing. Then the lower 3 cells, okay, out of the 5 or 7, the lowermost 3 cells of the female trichoblast will divide vertically. Okay, they'll divide up from down and form 3 tiers of the pericentral cells. Okay, so we had those 5. They will divide this way and it will form 3 tier, 1 tier, 2 and 3 tiers of the pericentral cells. Okay, I think if we look at the image, it'll be easier, no? Okay, see here. This is the female trichoblast. I want to pull it down. Okay, yeah. This is the female trichoblast or the short filament. Now, see, these are the apical cells. So, the initial doesn't start from the topmost. It will start a little bit down. So, there will be your initial cells, okay? So, these are the procarp initial or the carpogonial initial cells, right? Then, do you see they have divided and redivided and we form around 5 cells, right? And then we got your the mother cells, and the lower part, they will form the... Wait, is it going? Let's go up there again. Remember the vertical division? They will form the 3 tiers of the pericentral cell. Okay, there. I found the pericentral cell. Okay, first we started with just 2. Then we're having 1, 2, 3, 4, 5, 6, 7. Okay, so these are the pericentral cells. Okay, these were formed from the division of the lower cells. Okay, from the vertical divisions of the lower cell. Not that way. If this cell divide like this, that way, it will form the pericentral cell. Then let's go on again. Okay, so one of those pericentral, see here, one of those pericentral cells of the middle tier towards the mother axis will become the supporting cell. Okay, let me show you in the image. One of these pericentral cell, okay, any of these pericentral cell, they will become into this supporting cell. Okay, one of those. Now let's go up again. I think easier like this, no? Step by step I show you the image. Where were we? Then the supporting cell will cut off one small initial at its outside, which is the procarp initial. Okay, then that supporting cell will cut off one procarp initial on its outside. Okay, let's see again in the image. Okay, here. This is the supporting cell and this is the procarp initial. Okay. Then the procarp initially undergoes repeated division and will form a 4-cell branch, that is the procarp or the carpogonial filament. Okay, let's look again. There, you see that branch? This one, carpogonial branch, that one came from the procarp initial. Okay. See, this supporting cell did not divide. It stayed the same. But this procarp initial, see here I am circling, that one is dividing, and we found this carpogonial branch. Okay, so I think it's clear. Let me go up again. Then the topmost cell of the carpogonial filament will function as the carpogonium mother cell. Okay, so the topmost cell of that carpogonial branch, I think you can see it. This is those cells, no, from the procarp initial. This initial, it divided to form the carpogonial branch. Then this one, the topmost cell of the branch, carpogonium branch, will form the Carpogonium mother cell. Okay, then let's go on again. Then this Carpogonium mother cell will develop into the Carpogonium, okay, which is that flask-like, remember? So that Carpogonium has a swollen basal region which contains an egg and an elongated tube region called the trichogyne, okay. Okay, so that number is blocking. How do I? Okay, maybe like that. Okay, so this Carpogonial mother cell, I want this part to disappear. How do I do it? Forget it. Okay, so this Carpogonium mother cell will form the Carpogonium. Okay, it will just grow. This cell, no, it will keep on growing. Okay, do you realize? This will keep on growing and we will get this long tube, no, which is a trichogyne. And down there will be the Carpogonium, okay, which contains this egg cell. Okay, it will contain an egg cell. Okay, at the later stage, the Carpogonium will develop two initials from the supporting cell, one at the base, the basal sterile filament initial, and another in the side, the lateral sterile filament initials. Okay, so later on there will be two initial cells from the support, okay, from the down part. Okay, see there, from the support part, the supporting cell, remember, nothing was doing, no, the supporting cell was just staying like that, not doing anything, everything the other cell did. Okay, so now this supporting cell will release sterile filaments, sterile initials, one at the base and one at the side. Okay, let's look again. Yeah, one at the base and one at the lateral side. Okay, so even for these sterile filament to appear, they need one initial cell. So let's look at those initial cells. So remember again, we are talking about the supporting cell. Now from this supporting cell, from these supporting cells, okay, here it's very clear. From this supporting cell, no, you will get one at the base, which is sterile sterile filament initial, and one at the side. Okay, you see there, not one, there are two there. Okay, so you see two sterile lateral filament. Okay, and this one is one basal sterile filament initial. Okay, so this is how the development of the Carpogonium is occurring. Okay, now those lateral, remember, this is the supporting cell, this is the basal cell. There were two lateral, no, remember, lateral cells. Okay, so from these lateral sterile cell, initial cell, they will divide this way and form two-celled lateral sterile filaments. So that's why we see two-cell lateral sterile filament. Okay. Okay, let me say again for the supporting part. From this supporting cell, we will get one basal initial cell, one lateral initial sterile cell. Then again that lateral one will divide and will form two. Okay, so easy enough. Now this is final. Okay, this is final. So the Carpogonium is ready for fertilization at this stage. Then the pericentral cell adjacent to the supporting cell will start growing over to form to cover the fertilized Carpogonium. Okay, so because For the embryo, no, it needs proper protection, right, from the elements, from the environment. So it will it will grow a cover, then, or a sheet, okay? Just like in human, no, you have that placental wall lining, no, it's same, okay? So it will form a sheet around the fruiting body, and that sheet we call it as the pericarp, okay? It's just like, see, if this is the human, no, this is the human fetus inside, no, this one is the placental wall, no. So even here in the carpogonium, they will form a protective sheet around, okay, like a sheet. Okay, so I think if I explain from the diagram, it will be easier now, okay? Okay, so what is this? It is the short filament or the female trichoblast, okay? So from here, one of this cell will become the trichoblast initial,